Hello everyone! It's an honor to be presenting at this year's symposium. Thank you for having me. My name is AYESHA and I'm from Wild Oceans. My work and the project that I am here to talk about today focuses on the mesophotic reef biodiversity of the Comoros Archipelago using an ROV, which is a remotely operated vehicle. Okay, so these mesophotic coral ecosystems are classified by a depth range of between 40 and 200 meters. Mesophotic coral ecosystems serve as an essential habitat for some economically and ecologically important fish species, which use these areas for spawning, breeding, feeding, and growth to maturity. They provide refuge for coral populations due to favorable conditions in this less altered environment. They provide refuge from stresses such as extreme temperatures related to climate change or the impacts of fishing. These ecosystems are regarded as extensions of shallow coral ecosystems and share common species. As a result, scientists hypothesize that mesophotic corals may serve as potential sources to reseed or replenish degraded shallow water reef species. Coral reefs provide a multitude of benefits to human society. Mesophotic coral ecosystems contain organisms with specialized defenses to ward off predators and microbial infections. These specialized defenses often yield compounds that can be used to develop natural products and that benefit human health. And of course, they're used as a food source. So why are we doing this study? Why is it important? Despite the acknowledged value of these ecosystems, they are poorly understood and even more poorly documented. To date, 136 biodiversity studies on these systems have been conducted globally, with only 4 of these in the Indian Ocean. Without knowledge on where these ecosystems are, what comprises the communities, and how they change across geography and depth, it is difficult to designate, then appropriately manage, and protect them. In this study, we aim to document diversity of mesophotic benthos across depth and geography in the Comoros and explore patterns of these communities. Additionally, we aim to investigate specific component taxa of MCEs by documenting how octocoral communities change and the drivers of these changes. The overall objectives of the study are to improve the knowledge of the biodiversity of deep marine ecosystems in the Comoros Archipelago, improve the knowledge of fish species composition and abundance on deeper mesophotic reefs and the role of these reefs in sustaining inshore fisheries, to build civil society capacity in generating marine science knowledge and contributing to the conservation of biodiversity and the sustainable management of fisheries. An important part of this project has been and will continue to be community engagement. To share and discuss the project aims with leaders, managers, fishers, and tourism, and to train facilitators and teachers. Learning resources have also been provided to schools. And then, of course, to put that knowledge into practice, and hopefully this initiative will continue to grow. This is a map of our research area. The expedition focused on 3 islands of the Comoros, Anjouan, Grand Comore, and Moheli. These islands are found in the north of the Mozambique Channel between northern Mozambique and northwest Madagascar. At least 2 sites were sampled at each island. Moheli had 4 replicate sites, 2 inside the MPA and 2 sites outside the MPA. Video survey transects were collected at 3 different depth zones: the upper Mesophotic, which was between 40 and 60 meters, the mid Mesophotic from 70 to 90 meters, and the lower Mesophotic between 100 and 150 meters. And all this was done aboard the research vessel Angra Pequena. Surveys were conducted using an ROV equipped with a high-definition camera, lights, and two red lasers equidistant at 60 millimeters. Transects were run at each depth and focused on reef habitat. The ROV was kept between 1 to 2 meters from the sea floor during the transects with the aim of standardizing the area covered. A total of 30 ROV dives were achieved, accumulating approximately 50 hours of footage across all depths and providing 36 transects. This is an example of a transect image. It's in focus, not obscured by sedimentary suspension, and both lasers are clearly visible. These are the criteria that need to be met for an image to be used for analysis. 30 images were analyzed for each transect at each island at each depth, resulting in a total of 720 images. The analyses conducted utilized two types of software, namely ImageJ and Coral Point Count with Excel extension, otherwise known as CPCE. ImageJ was used for octocoral abundance counts, while CPCE was used to estimate percentage cover of the overall benthic organisms. The megabenthos, organisms clearly seen on the image and greater than 1 centimeter, were identified to the lowest practicable taxonomic level. Species-level identification often cannot be confidently assigned using images as specific characteristics require microscopic or genetic confirmation. Therefore, the majority of taxa were placed into putative morphotypes, that is, morphologically similar specimen. A total of 149 morphotypes were identified. This morphotype accumulation curve for Anjouan reveals that morphotypes stabilized after the 23 images, and therefore sampling effort was sufficient. However, an asymptote was not reached for all transects. Additional data are required to make robust conclusions. Therefore, 40 images per transect will be analyzed for the final results, which is the maximum number that fit the criteria. Across all transects, the number of morphotypes ranged greatly, between 37 to 60, with the fewest morphotypes present in the upper mesophotic zone. Crusto's coralline algae was the most common morphotype, and the richest morphotype group were the sponges. 31 morphotypes were documented in Anjouan, comprising 7 different families. Eliselidae and Plexauridae were the most common families, representing 18 of these morphotypes and the majority of depth specialists. This suggests that these families form the majority of mesophotic soft coral biomass in the Comoros. Eliselidae corals have previously been recorded over wide depth ranges globally and are considered to have broad ecological niches and function as depth generalists. Other generalist morphotypes had patchy distributions, so appear highly abundant though evenly distributed across transects. Though soft corals are not hermatypic reef building, they still provide structural complexity and have functional roles within the ecosystem. This graph shows the principal coordinates analysis, which provided a visual representation of similarities between communities at different depths. Here it indicates that depth is the major driver of megabenthic assemblage composition, with the transects from the lower mesophotic zone clustering together. Multiple morphotypes drove the difference between the zones. Example, the lower zone transects were characterized by hydroids, while the upper zone by hard coral and algae. Depth zones did comprise communities that were significantly different, and PEWA's comparisons also showed clustering revealed by the PCO. The preliminary data communities appeared to differ between upper and mid-mesophotic zones compared to the deeper zone. Data on octocoral alone mirrored this pattern, and this group also revealed an increase in depth specialists in the lower mesophotic zone. Here, the loose clustering of points implied dissimilarities between communities, though results show considerable overlap and within-group variation. The Permanova indicated depth was a significant factor in structuring overall community composition differences. And the pairwise comparisons between specific depth zones revealed that only the lower and upper mesophotic zones appeared to differ significantly. However, after Monte Carlo sampling and Bonferroni corrections, this was non-significant. For octocoral data only, evenness was similar across both depth and geography. It was also within a similar range as previously reported from other MCE communities. However, estimates of the richness indicated an increasing trend with decreasing depth, which was not expected and may be a result of undersampling. Further investigation, once all data have been collected, will help shed light on this. Communities of the MCE appear to be differentiated, suggesting the importance of the protection of these habitats across all depths. Further analyses will be conducted on the complete dataset, and the enumeration of octocorals from Grand Comore and Moheli will allow for a more in-depth study. The team will continue to work with taxonomic experts to assign identifications to morphotypes and indicate any geographic and depth range extensions. Abundance data will be used to conduct multivariate analysis, and these will be compared with presence-absence data. Stresses will be collated and applied to the biodiversity data in order to take note of any patterns. Together, these analyses will be used to help inform the prioritization of the designation of newly protected areas and will help to determine management plans for those sites already within the Moheli Reserve. Thank you.